Welcome to Lecture 8, Part 1 of Animal Cognition. Today we're going to turn our attention to memory. So we're going to begin by talking about what memory is, and then we're going to begin discussing the distinction between working memory and long-term memory, but the focus of this part is going to be on working memory and some of the methods that have been developed to assess the working memory capacity of individual species. So to that end, we're going to discuss radial arm mazes and the delayed match to sample task. And in the second part of this lecture, we're going to discuss a third method for measuring working memory capacity. So one of the things that we're going to see and explore is the fact that you get different assessments of working memory capacity depending on which type of measure you use. And we're going to examine why that is the case. But let's begin by discussing what is memory. So many of you have probably taken a memory and cognition course or cognitive psychology, so you've explored this topic in depth, so this should just serve as a brief review. Right? But memory in general is or you attribute memory to a species, whether it's humans or another type of animal. When you see some behavior change, when you see a new kind of behavior as a result of the animal having some experience. Right, so they experience something, and as a result of that, their behavior changes. And in general, memory is a cognitive process, and it's one that allows animals to adjust their behavior as a result of some experience. So from a cognitive psychology perspective, what's involved is an animal acquires some information, and then they apply that information in future circumstances. Right? So they store that information, right? They process it, they store it, and later they retrieve that information and use it in a new situation. And one of the amazing things is that memory can be seen in a wide range of species. And I'm going to try to give you a sense for this broad range that we see memory. But one of the things that binds all of this research together on animals and memory is the question of how similar animal memory processes are to those of human animals. Right? So if you've studied memory and cognition, you know that there's distinctions between types of memory systems. You see that there's certain phenomena, certain things that you see that are characteristic of human memory. And what researchers want to know is, do those things also apply to the memory systems of other animals? All right, so for example, does the multiple memory model apply in the case of animals? In the case of humans, it's common to distinguish between, for example, working memory and long-term memory. Those are two different types of memory systems that do different things, and we want to know whether those distinctions also apply to animals. So let's start with this issue, right? Working memory versus long-term memory. Working memory, which is also called active memory, is where an animal stores information that's currently being processed. Right? So it's where an animal holds information that's currently being used, and this information can come from either the environment, right? The animal sees something and then holds it in memory while they're doing some task. Or it can also, the information that's in working memory can also come from long-term memory. So they acquired some information much earlier on, like the location of a certain food source, and then when they want to find food, they activate this memory. They bring it forward to working memory, and they use it to constrain their behavior. Right, so that's the idea. Information goes into working memory, and it's used to guide an animal's responses. And one of the characteristics that we found in humans is that working memory is limited in capacity. Right? It's limited in how much information it can hold. It's often described as 7 plus or minus 2 chunks of information, right? Meaningful units. And another limitation is that it's limited in duration, right? Without rehearsing information, for example, information fades very quickly from your working memory. Long-term memory, on the other hand, stores information for extended periods of time, and it's not as limited in capacity, right? Your long-term memory cannot get filled up. So from an animal cognition perspective, long-term memory would be where an animal stores associations they have acquired, any facts that they have acquired, or events, right? Episodes that they've experienced, right? These things would be stored in their long-term memory. As I said, the memory for the location of food, for example, would be an example of a fact that they've acquired. All right, so that's a very basic distinction that applies to humans, and it probably also applies to animals. At least that's what the research tries to show. So I want to begin by discussing working memory and the different ways that have been developed to measure working memory capacity. As we've seen with humans, it's limited in capacity. So what is the capacity and how do you measure it across species? But let's begin by looking at an example of working memory in action in the case of an animal. One of my favorite species is the Portia fimbriata spider that we've talked so much about already in this course. As we've said, these spiders are aggressive and they feed on other spiders. And what they do is they invade the webs of other spiders to attack them. Right, so they like to sneak into the webs of other spiders, but sometimes they can't do this directly because they can be detected. Right, so often what they'll do is they'll take detours. Right, they'll go around and then try to sneak back around and then jump on the spider. But what's been observed is that sometimes the detours that Portia spiders have to take cause them to lose visual contact with their prey. Right? So as they're going around, they may lose visual contact, they may lose sensory contact with the resident spider. So what does that mean? That they don't stop, right? Just because they lose visual contact doesn't mean they stop and then they can't figure out what they're doing or why. Right? So the fact that they keep going around this detour, even though they've lost sensory contact, that's only possible if they've kept in memory, right, in some kind of working memory system, right, memory of its prey and also its location, right, so they have this goal that's active, that they're keeping active in working memory, and they're using that to guide their behavior even when they've lost sensory contact with their prey. Alright, so methods for studying working memory in animals. So it's been studied using what are called radial arm mazes, delayed match to sample tasks, and expectancy violation tasks. And the latter is the one that we're going to be discussing in the second part. But as I telegraphed earlier, right, not all of these measures produce the same answer as to how, what is the duration of working memory, right? They give you different answers for the same species of how long can animals store information for. So let's start with the radial arm maze. The radial arm maze was developed by David Olton in the 1970s, initially used to study rats. So these mazes consist of a central area with a number of arms that radiate out from this central area. And at the end of these arms is some food that's been placed there. So the animal, it starts in the central area and they have to run down each of these arms in order to get the food. So in order to be efficient at this, what they have to do is they have to remember where they have been so they don't keep going back to the same one. Right? That would be a waste of time and energy. And in the real world, that could mean, that they go hungry because other animals may take that food. So here's a visual illustration. You can see the central area, and you can see the different arms that radiate from it and the food that's been placed there. So how do you use a radial arm maze to test the duration of working memory? What they do, what the researchers do, is they put the animal in the central area. They let it run down the half of the arms, and then halfway through the task, what they do is they detain them. Right? They detain them for a certain amount of time, and they vary the duration that they're prevented from visiting the rest of the arms. So sometimes they force them to stay there by blocking off all the different arms, or other times they take them literally take them out of the maze and for a certain amount of time and then put them back in. So Beatty and Chevalier did something like this in 1980. They used an 8-arm radial maze with rats. The rats were removed from the maze after visiting 4 arms, and then they varied how much time passed before they were put back into the maze. And they varied it from 4 minutes all the way up to 24 hours, right before they were allowed to finish the trials. And what they found was when the delay was 4 hours or less, the rats performed perfectly, right? So they could recall which branches they'd already visited, and they didn't repeat those, right? So they knew exactly which ones to go down. But when the delays were 8, 12, and 24 hours, the rats started making a lot of mistakes, right? They started revisiting some of the branches that they'd already gone down. So what they say is that rats have a memory system for holding information for short periods of time. It lasts less than 4 hours, and they can use this to navigate the maze. Now, similar mazes have been developed for other species. For example, Spetch and Honig developed one for pigeons. Right, so they tested pigeons in a similar radial arm maze, and their delays range from 2 minutes all the way up to 120 minutes. And they found that the pigeons could handle delays of about 30 minutes. And after 30 minutes, they started to make a lot of mistakes, and their performance started to drop down to chance levels. So those are the radial arm mazes. They produce one estimate. Right, for rats, it suggests it's 4 hours or less. That's the duration of working memory. For pigeons, it's about 30 minutes or less. Another paradigm that's been developed to test the duration of working memory is the delayed match-to-sample tasks. And we've already examined match-to-sample tasks. Right, we've looked at this paradigm ad nauseum already. In a match-to-sample task, the animal sees a sample stimulus, and right away they see 2 choice stimuli or more, and they have to choose the one that matches the sample. Right? In a delayed matched sample, what happens is that there is a delay, right, a retention interval that's placed between the sample and the choice stimuli. So in order to perform accurately, the animal has to remember to keep active for a certain amount of time the sample stimulus, right? So, and what they to examine the duration of working memory, what they do is they manipulate the length of that retention interval. Once you've exceeded working memory capacity, then choices or working memory duration, the choices should be random, right? Should be chance. Right, so here's an example with pigeons. They see on the top, say a green light in the center, and then there's a retention interval where all the keys are dark, right? So nothing's going on for a certain amount of time. And then at the very bottom there you see 2 keys that are lit up on the bottom. Those are the choice stimuli, and they have to pick the right one that matches the sample. In this case, that would be the red key, the green key instead of pecking at the red key. And if they respond correctly, they get a reward. So studies using the delayed match to sample task produce very different estimates of working memory in different species, and ones that are very different from those that are observed using mazes. So for example, with pigeons, right, most pigeons show that they can only handle delays of about 5 to 10 seconds, and after that their behavior becomes chance, right, and that's when colored stimuli are used. Now Grant, a real trooper here, he found that he could get the retention interval, he could get working memory to remain accurate up to a minute with pigeons, right? But this required 17,000 training trials, which is ridiculous, right? Now, short durations of working memory are also found in other species using this task. With monkeys, for example, the maximum amount of a delay that can be used ranges from 2 to 9 seconds. With dolphins, they could handle retention intervals of up to 4 minutes, but still far shorter than the durations of working memory that you get with the radial arm mazes. So this raises a question, right? Why does the delayed match to sample task produce shorter estimates of the duration of working memory than does the radial arm maze? Well, there's 3 main reasons for this discrepancy in the estimates of working memory that you get, right? So the first reason is that the mazes, right, are very different from the delayed match to sample task in the sense that the mazes provide the animals with lots of cues, right? It's a very rich environment, right? And all these cues can help the animal retain where they've already been, right? And as a result, this could boost their performance, right? This could make them perform much longer. And another way of saying this is that the mazes don't just seem to be assessing working memory capacity, but they also seem to be assessing long-term memory capacity, right? Because long-term memory capacity involves using cues to try to extract something that's been held in much longer memory. Right? In contrast, delayed match to sample tasks, each trial is pretty much the same. Right? Pretty much the same. It's a very impoverished environment, so there's much more opportunities for confusion. It's much easier to get confused about this trial compared to the last trial. And another reason why you may get this discrepancy in duration is because the tasks are administered very differently. Right? The radial arm maze is usually done only 1 trial per day, right? While the delayed match to samples usually involve more than 60 trials per day. So again, more opportunity for confusing one trial with a previous trial. And finally, mazes are more ecologically valid since animals have to forage for food, right? So they have to do something that's much more natural to them, right? Animals are active. That's one of the main things that they do during the day, which is go scavenging for food. Right, so foraging is a natural behavior for more species, while the delayed match to sample task is much less ecologically valid. Right, it's not something that you regularly see in the wild. So that also may suppress their performance while the mazes increase their ability to perform. Alright, so we've discussed the distinction between working memory and long-term memory. We discussed 2 methods for measuring working memory, and in the next part I'm going to discuss a 3rd method for measuring and assessing the duration of working memory.